ये अभी स्क्रीन शेयर करूंगा, ये पीपीटी शेयर करूंगा तो फिर आप रिक्वेस्ट आप भेजोगे शेयरिंग
मैं
नोट्स का?
कोहोट कर रहा हूँ आपको, रुको एक मिनट।
हाँ
मल्टीपल प्रोजेक्ट शेयर अलो, ये है आपका,
हो गया।
हो गया ना।
ये हो गया। अभी क्या चलेगा?
लो, कोहोस्ट कर दिया आपको मैंने राजीव,
जी
आपको कोहोस्ट कर दिया मैंने। अब आप स्क्रीन शेयर के ऊपर जी करो,
ठीक है।
लाइव स्टार्ट हो गया, ठीक है।
ठीक है,
ये स्क्रीन शेयर पीपीटी शेयर करो, स्क्रीन शेयर हो रही है अभी ना, पीपीटी शेयर करो।
ये यहीं से कर लेंगे ओपन, देखो।
ये पीपीटी के ऊपर करके खोल दो, पीपीटी खोल दो पहले। एंटायर स्क्रीन शेयर कर दे, ऐसे करे राजीव।
हम्म।
पहले ये फाइल खोल ले, डेस्कटॉप पे खुली रहेगी ना ऊपर फाइल, फिर शेयर करियो।
अच्छा।
अब नीचे से जूम मिनिमाइज मिनिमाइज नहीं होना चाहिए, तो दाएं बाएं से खोल लो कहीं से फोल्डर ना, और वो फाइल खोल दो वहां पे। अब ठीक है, इसको
ठीक
बड़ा
है।
करना, बड़ा करना इसको। नीचे से स्लाइड शो कर दो ना, ये यहाँ पे, यहाँ पे ये नीचे से होगा, नीचे से नीचे होता है ये।
नीचे
हाँ, ये कॉर्नर बनाओ, इससे अगला, इससे अगला,
ये
इससे
वाला,
अगला, इससे अगला, ये वाला लेफ्ट वाला, अगला अगला,
ये
ये।
वाला स्लाइड शो।
हाँ करो क्लिक। हाँ अब ठीक हो गया, ऐसे करना, ठीक है।
अब
पीपीटी
इसको
जब
मैं क्या
चलाओगे,
करूँ?
हाँ स्लाइड शो पे पीपीटी खोल के रख लो, जो जिसने शेयर करने है ना पीपीटी, वो पहले ऊपर होनी चाहिए इसमें, नहीं तो खोलेगी नहीं फिर स्क्रीन शेयर में। ओपन करके ना उनको मिनिम ओपन करके मिनिमाइज कर दे, बाद में शेयर कर दियो उठा के, वहां से अपना शो कर देगा वो।
अच्छा ठीक है।
ठीक है,
ठीक है।
मतलब फाइलें ऊपर होनी चाहिए जब भी कोई चीज शेयर करो
हाँ
ना इसमें।
ऊपर है। हाँ
हाँ
ठीक है।
फाइलें ऊपर होनी चाहिए वो, क्योंकि फिर मिनिमाइज नहीं होता ना जूम, फिर दिक्कत आएगी। ठीक है, जिसकी पीपीटी हो ना, सब खोल के रखे, जो आएगा ले उसको शेयर कर दिए, शेयर करने के वहां से।
ठीक है।
ठीक है, ठीक है, चलो।
ठीक।
कोई दिक्कत है फोन कर लियो।
कौन है?
हाँ जी सर, क्या पी लो। ठीक, मेरी ना फाइल रही थी वहां पे बात सर के पास।
है ना। और ये बैनर वाला, ये वाला बैनर, ये दोनों की बैनर है।
जो भी आएगा यहाँ पे करके शेयर करेंगे।
अच्छा अभी ये वाला कर दो।
A very good afternoon to one and all present here. On behalf of CSR IMTech, I, Nandita Sharma, would like to welcome you all to lectures on the Nobel Prize in Chemistry 2024. It is being organized by Society for Promotion of Science and Technology in India, SPSTI, in association with CSR IMTech, chapters of NASI, INSA, and INYAS, CRIC Chandigarh, PI Rahi, and Amity University, Punjab, Mohali, and supported by Department of Science, Technology, and Renewable Energy, Chandigarh Administration. I would also like to extend a very warm welcome to our guest of honor, Professor Purnananda Gupta Sarma, Professor Biological Sciences, IISER Mohali. I request Professor Gupta Sarma, guest of honor, Professor Arun K. Grover, Vice President, SPSTI, and Dr. Sanjeev Khosla, Director, CSIR IMTech, to kindly take their seats on the stage. Thank you, sir. I request Dr. Sanjeev Khosla, Director, CSIR IMTech, to welcome our guest of honor, Professor Purnananda Gupta Sarma, with a flower pot and a shawl. I request Dr. Sanjeev Khosla, Director, CSIR IMTech, to also welcome Professor Arun K. Grover, Vice President, SPSTI, with a flower pot and a shawl. Thank you, sir. The 2024 Chemistry Nobel Prize recognizes the breakthrough in protein research, with David Baker from the University of Washington, Seattle, USA, receiving one half of the prize for computational protein design, and Demis Hassabis and John Jumper from the Google DeepMind UK for protein structure prediction as joint recipients of the other half. Proteins are essential biomolecules of a cell, enabling smooth functioning of various cellular processes in all life forms. Knowledge of the three-dimensional structure of proteins is critical to understand how these macromolecules, which have evolved over several billion years, perform various functions. With that, I request Dr. Sanjeev Khosla, Director, CSIR IMTech, to formally welcome our audience. So please.
Very warm welcome to all of you who have come from outside IMTech and from within IMTech. Warm welcome to friends Purnanand and Dr. Grover, and from SPSTI, all the dignitaries here. It's indeed a pleasure to actually be part of this program because, as Nandita said, this is a very important area and something that IMTech also has been working on for a very, very long time. Today, two of our scientists, Dr. Shri Krishna and Dr. Beena, who have been working on protein biology, would be talking about the Nobel Prize that was given this year in chemistry. So I'll not take too much of your time. Welcome, warm welcome to all of you. And I'm very sure we'll all go back with a lot of knowledge from these two talks. Of course, Purnanand would also be prefacing all about the Nobel Prizes today. Thank you very much.
Thank you, sir. I now request Professor Arun K. Grover, Vice President SPSTI, to give opening remarks and brief on the lecture series. Sir, please.
So on behalf of SPSTI, Chandigarh chapters of Science Academies, INIAS, Amity University, Punjab University, CLIC, Pai Rahi, and other institutions, I'm very happy to welcome all of you to this year's Nobel Prize in Chemistry. So we started hosting these Nobel Prizes in the 4 science subjects, plus that in peace and literature 5 years ago, and this is our 5th year in running. All the lectures are available, all the previous recordings are available to you if you just go to the SPSTI site. So purpose was to create a resource for our community, so people can go and learn about these whenever they want at the time of their convenience. So for 3 years, we had these lectures in the online mode, but this year, our president, SPSTI, Mr. Dharamveer said, no, we must attempt to do it in a hybrid mode. So we did few in the hybrid mode last year, but this year we are doing all 6 of them in the hybrid mode. So this is our 3rd lecture. Physics was to happen last week, but the speaker had some issues. So it is now postponed a little bit. And the 2 more on economics and peace, they will be via evening lectures in the hybrid mode done at IISER Mohali in 2 weeks from now. At least 5 out of the 6 lectures we hope to complete by December 10, the day of the Nobel Prize ceremony in Sweden. So I'm just tempted to recall the visit of Venki Ramakrishnan to our region in the year 2016. He gave 4 public lectures, 1 at PGI, 1 at Punjab University, 1 at MTech, and the 4th one at IISER Mohali. And I am also tempted to recall how Girish was excited when the G.N. Ramachandran Center had been opened by none other than Professor Balram at MTech. So you have a resource which the younger people in the city can come and visit and learn about it later. And I am told by the organizers that they will open a visit to the G.N. Ramachandran Center for the other for young people from other institutes to visit this year as a part of commemoration of Nobel Prize in Chemistry this year. Thank you very much.
Thank you, sir, for that great opening remarks and all the flashbacks of the various lectures that have been held. And with that, I request Dr. Nishima Wangu from Punjab University and CRIC, member of INIAS, to introduce our guest of honor and request him for a welcome address and address the gathering.
Thank you so much. I'm Nishima. I'm a faculty member at Punjab University and a member of Indian National Young Academy of Sciences, INIAS. And I have been given this pleasant duty to introduce Professor Poonananda Gupta Sharma sir. And although for the IMTech family, he needs no introduction, but for those who are from outside IMTech or from outside IISER, I would like to give a brief introduction about him. Professor Gupta Sharma has been working as a professor at the Department of Biological Sciences, IISER, since 2010. He worked as a scientist at IMTech also from 1996 to 2010. He has held many administrative positions, including Head, Department of Biological Sciences, IISER Mohali, Dean R&D, Coordinator Technology Business Incubator at IISER Mohali, its member of Board of Governors, and Dean Faculty Affairs. He won the Tata Transformation Prize in 2023, AVRA Young Scientist Award in 2005, CSIR Young Scientist Award in 2000, amongst many others. His research interest is mainly in the area of protein engineering and structural biochemistry. He has many scientific honors to his credit and has also rendered many scientific services. He has supervised nearly 23 PhD theses. I'm sure many are in the pipeline also, and has many patents and publications to his credit, and also books and book chapters. He also has various extracurricular interests, as for example, music. We have seen his posts on YouTube also. Sir sings really very well, and in reading, writing, etc. So with these few words, I would like to welcome Professor Gupta Sharma, and I would like to ask him to please come over here on the dais, please, sir.
Just plug it in. Can you help me?
Yeah, they will help, sir.
Okay. So while they set it up, it's a pleasure to be back at IMTech. Every time I come, I've spent 14 years here and I've spent 14 years at Isamawali, so it's exactly half an hour. Sanjeev said that I will introduce the Nobel Prizes. Actually, that's not what I'm trying to do. Krishna and Veena will talk about the Nobel Prizes, and I will present the background that you need to begin to feel so that you appreciate what they say better. Because if you do not appreciate why protein folding is interesting, you will have trouble, I imagine, understanding why the Nobel Prizes were given. So let's try, let's see which one works. So first, what I'll do is try this and see if the movies work. I have a few movies. So let's see if one movie works. Yeah. Okay, great. Thanks. I'll use this.
making the share कर देता हूँ सभी.
Okay. Let's go for full screen. So the two Nobel Prizes have been given for computational determination of the structures of proteins from their sequences. And I'm going to talk about why that is such an amazing thing, why it could not have been done by any method other than recombination. So I thought I could title my talk in two different ways. The first is what it says here, a few words about proteins and protein folding, for those who don't commonly think about these things. And the second is
left, right.
thank you. This is for the pointer, and this is forward and back. So the alternative title for the slide is why protein folding could never have been addressed computationally without artificial intelligence. I'm an OIC. OIC, okay. Is this not on? It's nearly 3:25. I was told I have 25 minutes, but you can always cut me short. Okay. So we have known since about the 1990s that protein folding would eventually be addressed by artificial intelligence through computation and not by any other methods. And we've been waiting for decades for raw computational power to catch up and for abilities related to parallel processing in computation to catch up. And it's finally here. I think 2 or 3 years ago, AlphaFold first began to predict the structures of proteins from sequences. But it's good to start at the very beginning, which is usually a very good place to start. I didn't get any laughs. That means people here haven't watched the movie Sound of Music, which has a whole song called Do Re Mi, which begins with these words. Start at the very beginning, which is a good place to start. For some reason, the things written at the top of the slide are not showing. Is there anything that you would like to do? I click on the OK. Okay. So proteins are long chains of amino acids. What you see here on the slide is an amino acid. The reason it's called an amino acid is because it has an amino group here, an NH2, which can turn into NH3+, depending on the pH, and an acid group here, which is a CO minus, which can turn into COOH, again, depending on the pH. There's a central alpha carbon atom here, a proton to one side, and something called the side chain represented here by R, which is off on the other side. Every amino acid that you can think of has this structure, an amino group and an acid group, and a side chain. And of course, the side chain can be anything you wish it to be. But in nature, there are only 20 naturally occurring amino acids out of all the conceivable side chains that you can have, which would define different amino acids. And these can be subclassified into aliphatic, tiny, small, polar, positive charge, non-polar, aromatic, and so on the basis of their size and character. In proteins, these amino acids are strung together into long chains by peptide bonds. I'm not yet familiar with this pointer, so I'm making some mistakes. By peptide bonds, and these chains can differ in length as well as in amino acid sequence. So this is a chain here shown as a cartoon with these side chains popping out in different directions, alternate directions in this case, each being of a different shape, showing that every amino acid here along the chain is different. And what you see here in this panel on the right bottom is the fact that you have a lot of sigma bonds and some sigma bonds that masquerade as partial double bonds. In particular, the peptide bond between amino acids has a 40% double bonded character. But there are many rotations around these bonds that are possible, which allow the atom groups flanking these bonds to rotate. So the stringing together of these amino acids is done by ribosomes. And once that happens, these chains have to transform themselves from these extended polymers into unique, compact, three-dimensional shapes. And for that, these chains have to undergo folding. So they undergo folding to give rise to a unique structure, which we know of as the native structure of a protein. And for this to happen, amino acid side chains have to pack even as they fold. What is shown here is a very small polypeptide that is undergoing this process of folding. But this can happen for chains that are as small as 50 amino acids in length and as large as 6,000 amino acids in length, except that if they're very long, there would be sections of chain which would do this independently of other sections of the chain, and the whole 6,000 amino acids would not just form one large structure. So amino acid side chains pack as the protein folds, and they generate a microparticulate solid, which you can see here, that's the solid, right? Which has a unique and defined structure. And this solid is chock full of atoms. It's almost as if it's fitted together with optimal packing, as if it were a three-dimensional jigsaw puzzle. So you start with something that is sort of quasi two-dimensional, and it folds through many, many influences that operate between the chain and its environment. And then it folds into a unique three-dimensional structure, which seems to have very, very optimally packed side chains. And these structures are describable at multiple levels, which are called primary, secondary, tertiary, and quaternary structure. The primary structure is just the definition of the sequence and any other covalent bonds that exist, such as disulfide bonds, for example. The secondary structure describes the hydrogen bonding networks that create helices, sheets, and other kinds of structures which are regular structures. The tertiary structure describes the coming together of secondary structures of different types into a unique three-dimensional shape for a single chain. And a quaternary structure describes how multiple chains, shown here, for instance, in this diagram with different colors, come together to form a multi-subunit protein. In this particular case, what is shown is a human hemoglobin molecule. What is shown here on the left is a ribbon diagram representation, which simplifies our ability to understand what the structure of the protein is about. Whereas in reality, all along this ribbon diagram representation, there are side chains. And so the real hemoglobin tetramer actually looks like this when you look at a space filling model. And this process called protein folding generates a very diverse group of tens of thousands of different molecules. I say tens of thousands because in bacteria, you have a few thousand different proteins, and in humans, you have about between 20 and 30 some say about 24, 25,000 proteins. But if you look at the proteins of different organisms, some of them are the same, not all of them. And so we can say tens of thousands of molecules with three-dimensional structures that are perfectly formed and which are exquisitely adapted to the performance of the functions that those proteins then perform. So thus, we can say that folding is actually critical for protein function to occur, and proteins are the workhorses of living cells and of living organisms. And so they are the fundamental elements of life which use information that is present in the nucleic acids to create themselves, to create nucleic acids, and to perform all kinds of functions through the recognition of other proteins, of small molecules, of nucleic acids, and what have you, and to also catalytically create a whole bunch of molecules. So proteins lie at the heart of all life, and if these proteins didn't form through folding, there would be no life. So that is a very nice context to present for a talk such as this by way of introduction. The next thing that I want to say is that many years ago in 1972, someone called Chris Anfinsen got a Nobel Prize for protein folding, and this year's Nobel Prize in Chemistry is the next one in protein folding after that. And what Anfinsen showed is not what is shown in the rest of the slide. I'm just summarizing it in one sentence. What he showed is that amino acid sequences specify three-dimensional structure. So for every protein, its amino acid sequence determines its three-dimensional structure. And the way he showed that was very interesting. He showed by taking a protein called RNase A, breaking all the disulfide bonds in it, causing it to unfold, and then allowing it to refold in solution, and obtaining a very high efficiency of refolding of the population to native structure, to an active and functional native structure. And so that showed that in the absence of any helping molecules such as chaperones and so on, the information for the formation of the three-dimensional structure of that enzyme was present in the sequence of that enzyme. So this is what Anfinsen showed, and we have later learned from studies of the sequences of many, many different proteins that similar sequences hold to form similar structures. So for instance, this here is triphosphate isomerase shown as a representative example. The first sequence here, which goes from the left of the top line to the right of the top line, and then again in the bottom panel from the left of the top line to the right, is for E. coli triphosphate isomerase. It's an enzyme that participates in glycolysis, a very important enzyme that's present in every cell of every living organism on Earth. And what you see below that are the sequences of triphosphate isomerase from a variety of other organisms, going from bacteria through yeast and thermophilic bacteria to birds, and then to plants, humans, and hyperthermophilic archaea, and what have you. And these sequences have many commonalities. They have similar lengths. There are many places along the sequence where the amino acid that is used is exactly the same. And these are shown by these colored lines that run from the top to the bottom. Wherever a colored line runs all the way, it means that those are the positions at which the sequences, the amino acid identities are exactly the same. And these seem to be the core positions where amino acid identity cannot be allowed to change if the chain is to fold into a given structure. What I'm trying to emphasize through this slide is that these are the conserved positions, but there are other positions where a lot of different variations are allowed, and these variations are compensated by other variations elsewhere in the chain, so that the overall structure remains conserved, while little bits of it in the form of the sequence are allowed to vary. So eventually you have a whole bunch of proteins, and although they're all very similar in that, just like different animals have, radius and ulna and, humerus and all of these other bones in terms of skeleton, all proteins have alpha helices, beta sheets, beta turns, and other such structures, which form its skeleton. But once you decorate that skeleton with the flesh and the muscle and the blood and everything else, which I'm referring to in terms of the side chains that you decorate the backbone with, then you get a whole lot of different proteins performing different functions. And therefore, many different proteins have to be studied in many different ways before any general inferences can be drawn about the range of their structures and their behaviors in solution. And proteins are also not necessarily static. They're actually extraordinary machines, which in this case, what you see very briefly here, because I think the CPU of this computer is very fast, is a little dot here. which is a metal atom that comes and binds to the protein. And as it comes and does that, the protein undergoes a conformational change that is a part of the function that it performs. And this conformational change occurs cyclically with every round of catalytic activity. So the native structure, which is necessary for the performance of that function, is the only structure that is ever apparently stably formed by the folding chain of a naturally occurring protein. Of course, there are some exceptions where there are alternative structures that are possible. And what is really, really interesting, and this is what is the core of the protein folding problem, is that it's not the only conceivable structure out of all structures, right? So proteins fold to the native structure, and that's exactly what is so surprising, that they fold into a single native structure at all when there is a virtual infinite number of structures that each protein can potentially fold into. Now, for those who haven't thought about this, it's necessary to show a few slides to emphasize why this is the case. So you see, along the backbone of a protein, 95% of the chemical bonds are sigma bonds, right? Which are single bonds. Of course, the peptide bond is a CN bond. It should have been a sigma bond, but it has a partially double bonded character because two lone pairs of electrons around the nitrogen and the oxygen move into the peptide bond. So they give it a partial double bonded character. But otherwise, there are enough sigma bonds in the chain, both in the main chain, in the backbone, and in all of the side chains, because of which we can say that more than 95% of the chemical bonds in a protein are sigma bonds, which allow rotation of atom groups that flank them, right? And the rotations are shown in some of these pictures below. What I'm going to show you next is a visual, some sort of visual imagery where what I show here is just two carbon atoms and then three other atom groups that are there on both sides, let's say occupied by different atom groups. So one could be just a proton, one could be methyl, one could be ethyl, so that they're not equal or not equivalent. And then in this movie here, which I think I'll have to play with the mouse, and I'll do it. So you see, that's one rotamer, that's another rotamer 120 degrees away, and a third rotamer 120 degrees away. And if you do it thrice, you come back after that to the same rotamer, right? So let us assume that although what I'm showing you here is supposed to be a CC bond, right? Because I've shown two metal spheres there that look similar. Let us assume that these are any two atoms, right? And around those atoms, you can have rotamers forming. So we can summarize that by saying, don't look at the title first, just look at what I've said here in the main text. Assuming conservatively that each sigma bond has three rotameric states, the three rotameric states that I just showed you using that video, that animation or whatever, and that each amino acid has at least three sigma bonds. This is very, very conservative because along the backbone, each amino acid has at least two sigma bonds around which rotation can occur. And we can assume that the side chain has at least one. In many cases, it has 6, 7, 8, 9 sigma bonds. So this is a back of the envelope type of calculation that I'm doing on this slide, just to say that every amino acid has at least three sigma bonds around which rotation can occur. If there are three rotamers around each sigma bond, then a single amino acid should be able to adopt 27 different structures, 3 to the power 3. Therefore, if you have a 100 amino acid long protein, this should be capable of adopting at least 27 to the power 100 structures, because for every position you have 27 structures, and you have 100, so that's 27 and 27 and 27. So you have 27 to the power 100 structures. And do you know how big is 27 to the power 100? Just for comparison, I've put up in the next slide two figures which will tell you how big it is. So the number of atoms in the observable universe, which I've written here as approximately 100 billion galaxies of 100 billion stars each. Some people now say it's actually 100 trillion galaxies of 100 trillion stars each. These numbers are changing as you have better and better telescopes. So the calculations that the physicists do, and we have a very eminent physicist sitting with us here, Professor Grover, and Professor Kea. Professor Bhasin, you're a chemist, right? There's not really much difference between physicists and chemists. Okay, it's estimated to be only around 14 to 10 to the power 78 atoms in the observable universe. Now, if you look at the number of femtoseconds, a femtosecond is a millionth of a second, for which the universe is thought to have been in existence since the Big Bang occurred, about 14 billion years ago, approximately, is only 14 to the power 32. And what is the number that we're talking about? 27 to the power 100, which is also a very conservative estimate, assuming only 3 rotatable bonds per amino acid. So the number of structures for a single chain encoded by any one gene in your genome is humongous and unimaginably large, right? And that's not the only thing that's unimaginably large. Here's another thing, right? Which is there are 20 different amino acids. So each of the 20 can be the amino acid residue that occupies any position in a protein chain. And so if you have a 100 amino acid long protein, you should have 20 to the power 100 different sequences that are 100 amino acids long. So 20 to the power 100 is not a whole lot different from 27 to the power 100 from where we are sitting, right? Both are large infinities. So from an infinite number of possible sequences of just 100 amino acids, and remember proteins can range from 50 to 6,000. From an infinite number of possible sequences that nature could have picked, nature has picked only a few. How many? Without defending it, I'll tell you about 10 to the power 14 sequences, right, of all possible lengths. 10 to the power 14 out of 20 to the power, not 100, but 6,000, because that's the largest protein, right? And so, if you say, okay, whatever you're saying is a percent of something, then there'll be only a change of 2 in the exponent, right? So it's a long way to that infinity. What has caused nature to pick these particular sequences out of an infinity? And then for each sequence, you have an infinity of structures that can be adopted. What has caused nature to pick one structure out of that infinity and then use that as the native structure? And how in heaven's name, I was going to say hell, but one should not say hell, apparently. So how can nature have programmed every one of these sequences to have folded to a native structure? But without worrying about how, one can worry about what, because there are always all these questions in science: what, why, how, when, where, how, and so on, right? How much? So without worrying about the how, one can worry about the what and say, okay, I don't care how complex the problem is. Is there a solution? This is typically the difference between a scientist and an engineer, I found, because I happen to train in both, while doing a dual degree at BITS Pilani. So scientists love insights and engineers love solutions. So actually, the Nobel Prize in Chemistry is an engineer's fantasy, because what has been found is solutions, which Krishna and Veena will tell you about. Insights are not that many. Artificial intelligence never provides insights, because these are like black boxes, engines that give you answers, but they don't tell you how they arrived at the answer, and they can't actually explain to you how they arrived at the answer. Let me now just do a quick check of 25. So I finished 16 minutes. I have another 8. Let me see if I can very quickly go through the rest of what I want to say. So every copy of a protein of a particular sequence in a solution, this is just a summary of whatever I've said. Every copy of a protein of a particular sequence in a solution is chosen out of an unimaginably large number of sequences that can potentially exist. And it has to adopt one structure, which is the native structure, out of an unimaginably large number of structures that it can potentially adopt. How does this happen? And how does one compute it if the information lies in the sequence itself? So the very first attempts at computation use brute force methods of using force fields, calculations of interactions between charged residues of opposite character or similar character, which are attractive or repulsive, and what have you, right? And they tried to predict structures by taking a chain, predicting every possible structure. How many? Infinite numbers of structures. Predicting every possible structure and calculating the energy. That's something you can do very easily. Take a particular structure, just rotate one of the bonds in the structure by, let's say, 30 or 120 degrees, and compute the energy. Then change something else, compute the energy. And do this for every bond, every rotamer, both in the main chain and in the side chain. Do it for an infinite number of structures, tabulate the whole thing, write all the energies, and then see which is the lowest energy, because somebody hypothesized that the native structure will be the structure of the lowest energy for chemical reasons, right? For reasons of thermodynamics. Now, how long would it take to do that? So a petaflop per second capacity machine would take 3 years to simulate just 100 microseconds of dynamics of a short protein. That means the protein is doing its own thing in the solution, right? and you're simulating the dynamics of that just for 100 microseconds, which is a tenth of a second. Just to do that, you would take 3 years using a petaflop per second capacity machine, which was not even available, right? So these are the sorts of computational timescales for protein folding. There are a lot of details given here. But the point I'm making, I can just go to straight away and say that the lifetime of the universe would be insufficient for any ordinary, computational solution, because even if you made something that computed the energy of a particular structure every femtosecond, and the next femtosecond it changes the structure, computes another energy, in the next femtosecond it does so and so on. In all the time that has passed since the Big Bang, for one protein sequence, you would end up tabulating only an infinitesimally small number of structures and energies, right, out of all of them. So this was not a computationally tractable problem. People realized this in the 1990s and said, we have to do it through artificial neural networks, which had already been invented. Many of the algorithms for artificial intelligence were invented 40, 50, 60, maybe 70 years ago, right, mathematically. It's just that we did not have the raw computational power. Now we have a huge amount of computational power, parallel processing, high performance computing, cloud computing, and so on, right? And also all of those old algorithms now are able to do what Krishna and Beena will tell you about. They were like sitting like this. I hope he's not going to steal our thunder and talk about it right away. No, I'm not. I'm not saying anything about the Nobel Prize. I'm only saying why the Nobel Prizes are so interesting. There's a noise coming from somewhere. Oh, it's coming from the video. Right. Oh, yes, of course. Can we switch off that sound? No, maybe not. Okay, so now you see, things would be easier if proteins behave like elastic objects that are capable of remembering their structures. Switch it off so that I can just run it. Yeah. So it would be easier if proteins behave like elastic objects, right? All know what an elastic object is. So you saw these videos. In one, there's a telephone wire. The young people here don't know what telephone wires are because landlines have disappeared, but the older people do. So these are these spiral helix-like structures. If you pull them and let go, they reform their old structures. They're elastic, right? The material remembers its structure once it is formed. On the right also, what I did is to just tear off a piece of sponge, and it's an irregular piece, right? And I just compress it into some random shape, right? And let go. This is a proxy for unfolding and refolding. And I let go and it remembers the structure. So if proteins were able to do this, it would be very easy to explain Anfinsen's Nobel Prize. Because what Anfinsen showed is you unfold a protein and let it fold, and it refolds on its own. So maybe it's an elastic structure which remembers its structure, so you can do it any number of times. Unfold it, let it go, it refolds. Unfold it, and it refolds. But, and nothing could be more critical than this slide that I'm showing you, proteins are not elastic materials. Okay? They are plastic objects with many degrees of structural freedom, and they cannot remember their original structures. Once the structure is destroyed, the information necessary to recreate is no longer present. Each molecule has to work it all out each time it undergoes folding, and usually by itself because there are no chaperones or other helpers present, right? And I'm going to show you a movie that I made many years ago, which I hope you remember, but I should give you a background to this movie. And I'll stop at 25. So what's shown in the movie is the sort of red string that some people tie around their wrist, when you go to a temporary flight. So I took it off one of my students' wrists, asked her to take it off, and then put gum all along the string. And I should go back to that one. So I took it off, took off this string off somebody's wrist, and put gum all along the string, and then put it out in the sun to dry, and then let it dry, and then compressed it into a ball, seeing to it that the two ends would remain visible. And then, so imagine this is the protein, and the two ends are the N and C termini. And now, using something, breaking hydrogen bond, breaking electrostatic interactions, breaking hydrophobic interactions, or just pulling at the two ends with optical tweezers, I unfold the protein, right? I unfold the protein. Yeah. Now. Okay. Very good. Are you getting a good thing on Adnan? Yeah. Excellent. Good. Excellent. Open it up, open it up, open it up. And just leave it on the. Okay. So this is a string that has been unfolded, a gummed string that has been unfolded. Let's say that I ask another student to pack it back into a sphere. Would they be able to do it? Yes. Why? Because there is gum, right? But would the sphere be the same sphere as the earlier sphere? Would the trajectory that the chain takes through that sphere be the same as in the earlier case? The answer is no, right? It's evident to anyone who thinks about it, right? So there are sticky interactions in proteins. There are hydrophobic interactions, hydrogen bonding interactions, electrostatic interactions, and other favorable van der Waals interactions, as well as a lot of unfavorable van der Waals interactions, right? And all of them create a metastable structure for the native structure, which once you destroy it, there's no information, apparently, because any CO can hydrogen bond with any NH, any aspartate can form electrostatic interactions with any lysine, any leucine can form hydrophobic interactions with any tryptophan, and in any case, all of these interactions only happen at very short distances. Hydrogen bonds will happen at 2-3 angstrom distances. Electrostatic interactions are useless beyond 3-4 angstroms because the Coulombic law dictates that as you go further away with the second square of with the square of distance, the interaction drops. So the point is, when you understand all these interactions that stabilize the folded structure, the native structure, what you're understanding is what holds the structure together once it is formed. What you are not understanding is how does the structure form in the first place? What causes the chain to fold in such a manner that all the things that need to interact are at angstrom distances away from each other so that these interactions can occur, which you see in the native structure. So all of these interactions are ultimately useless for predicting structure because many of these interactions have high affinity, but very poor specificity. Okay, I've made the most important point that I want to make, so I could just stop, but I'll go on to 5. I have just finished 25 minutes, so I'll just wind up. So proteins are much more like the gumball strings that I showed you and much less like the telephone wire or the sponge. And although many different kinds of gum are used, all of these interactions, they provide affinity, but no specificity, right? And so folding absolutely challenges credulity. If there hasn't been enough time in the universe to physically sample all of the possible conformations of a given small protein of 100 amino acids, when did nature have the time to evolve so many different protein sequences that fold so efficiently to different structures? And when did nature have the time to also discover the pathways by which to make these sequences fold so rapidly? How rapidly? In some cases, in milliseconds. Those of us who work with these in the lab know that sometimes the whole population folds on a millisecond time scale with 95% efficiency. That means 95% of the unfolded molecules actually reach the native structure. But where do they start from? So I'm not going to explain all this, but I'll just leave you with 2 or 3 slides. One is this slide, which is a 2-dimensional representation of what appears to be a 3-dimensional graph, which is actually multidimensional, right? And every point here on this plane represents the unfolded state. And you can see there are many different unfolded states, right? Represented by 6 or 7 conformations, actually by as many conformations as there are molecules, which is very easy to understand. For instance, suppose the number of chairs in this room is larger than the number of people who have come into this room to sit. What is the chance that any 2 people will sit on each other's lap? 0, right? Because as long as the number of states is larger than the number of objects trying to occupy that state, no 2 will occupy exactly the same. And so if you have 10 to the power 17, 10 to the power 18, 10 to the power 19, 10 to the power 20 molecules in your experiment in solution, and you have 27 to the power 100 or 27 to the power 6,000 structures to adopt, what's the chance that 2 unfolded chains will have exactly the same structure? Zero. Starting from very, very different structures, therefore, they have to fold to form a single structure over a time scale of a few milliseconds or seconds, and sometimes minutes, sometimes hours. I've worked with proteins that fold on a time scale of milliseconds as well as that fold on a time scale of days. How does it all happen? Days is also very little time for all this to happen, right? So there are 5 incredible things about protein folding, which I would have explained to you in the slides to come, but I would be the last to be accused of eating into Sri Krishna's and Deena's time. So I will just leave you with these 5 without explaining them. Each had a slide to go at the end. Folding occurs at an incredible rate that we don't understand. There's incredible amounts of information encryption which we don't understand, both in the form of folding fate, what the final structure to form, and in the form of folding pathway, how to form that structure. One pathway, multiple pathways. Efficiency is incredible because 95% of some proteins, 90%, 70%, 80% of a population can fold. There's incredible variety in sequence and in energy landscapes, and there's also incredible selectivity. I will leave you with, okay. I hope one of you might talk about thermodynamic control and kinetic control. I don't know whether you're going to, probably not, okay. This would have been a really interesting thing to explain, but I will not. So I'll leave you with the concept of folding intermediates, that they have to be present as you go from a multiplicity of unfolded states to a single state. Of course, there has to be a narrowing funnel of the number of structures that can be adopted. And so how does all of this occur? With how many types of intermediates is anybody's guess. And what the Nobel Prize has done is to leave us unsatisfied in respect of all of these questions, right? What it has done is to use, I'm sorry, that should have been big data analysis, not big data analysis, and neural networks to see patterns that we cannot see and to predict structures from sequence alone. And what patterns do the AI routines learn? Unfortunately, they cannot tell us what they learn. So I will predict, and let's see what, just to make this interesting, what Beena and Sri Krishna have to say about that. I will predict that what they learn from looking at sequences and comparing structures and doing all this big data analysis inside a black box, right, is that they learn to simulate de novo folding. That means the folding of the type in which the N-terminal regions of the sequence are much more important for determining what happens than the C-terminal regions because they come first. Just like in a family, the eldest sibling is the most important in determining what happens to the next sibling. So the first domain to fold, the first subdomain to fold, and so on. So this is what I feel that this is what AI routines are picking up as patterns, and prove me wrong. I'm sure nobody can, because we don't know what those AI routines are really doing, but I'd love to learn what they're doing. The end of the taste of the pudding is in the eating, which is what the engineer says, right? Solution rather than insight. So from that point of view, AlphaFold and all its versions have been terrific. If they actually all work, that data is also not in. Personally, I feel these Nobel Prizes are a little premature, but I'm very happy that they've been given because they recognize an area that I'm very interested in. And that is always very convenient. For example, I had the opportunity to talk to all of you distinguished people just because the Nobel Prize was given for protein folding by artificial intelligence. Thank you. So the reason that I gave an address, which was actually like a scientific talk, is because I was asked to. I was told that you're guest of honor, but please give a talk like the others, right? Maybe pen drive, lovely. Thank you.
Have you kept it here? Yeah, it'll be this side. Thank you.
Thank you, sir, for that insightful lecture. And indeed, now we know why it's always good to start from the start. Now, once again, I would like to request Dr. Nishima Wangu to introduce our first speaker for today's lecture series.
Now I would like to welcome Dr. Sri Krishna Subramaniam. He is currently Chief Scientist at CSR MTech here. He obtained his PhD in 2001 from the Molecular Biophysics Unit, ISC Bangalore, where his research focused on macromolecular X-ray crystallography. Subsequently, as a postdoctoral researcher, he honed his skills in computational structural biology and made significant contributions to understanding protein evolution. Over the past 2 decades, first as an independent researcher at the Burnham Institute for Medical Research, US, and then at IMTech, his research has focused on classifying protein structures into evolutionary meaningful families and developing novel algorithms to assist in this process. This work has contributed significantly to understanding the mechanisms of gene and protein evolution and the roles of these macromolecules in their respective organisms. Dr. Subramaniam has published over 125 papers in reputed peer-reviewed journals, and he's also on various boards as task forces, as members of task force committees of DBT, SERB, NPDF, ICMR, and many others. So, with this brief introduction, I would like to welcome Dr. Subramaniam. Sir, please.
शेयर कर कितने शेयर कर दो
Screen.
नहीं कौन सी है अच्छा ओपन कर देता हूँ
Welcome all. I would like to thank the organizers for giving me this chance to, speak about this year's Nobel Prize. So, this year's prize in chemistry goes to a subject which is very close to my heart, just like what Ponanan was saying. It has been given to 3 people. And this is incidentally also the first time AI-based method has actually got a Nobel Prize. So the prize was given to David Baker for design of proteins, which Beena will cover. I will cover the other half of the prize, which was jointly given to Demis Hassabis and John Jumper, who are both from the company DeepMind, which is part of Google. Demis has a very interesting history. He is a chess player and also a game developer. And this is something for kids who are here. You don't have to always be studying. You can also play games and get a Nobel Prize. And he started, he did a PhD in neurosciences, and then he started this company called DeepMind in order to try to understand intelligence computationally and then use it to solve other problems. That is the motto of DeepMind. John Jumper is a traditional biophysicist turned chemist whose interest is in proteins and simulations. And both of them are very young people who have got this prize. So Purnanand has done, made my job very easy by, giving you a very thorough introduction of proteins. But if you look at the building blocks of life, it's not just the proteins. Proteins are very important, but then there are also, nucleic acids, carbohydrates, lipids, and so on. And the central dogma is that the information that is coded for in the nucleic acid of organisms, what I show you in this diagram here, codes for the primary structure or the protein sequence. This knowledge that you can actually go from the DNA to RNA to the protein sequence, one, Hargobind Khurana and Marshall Linenberg, the Nobel Prize in 1962 for trying to understand what is the code that actually takes you from the DNA, that is the information thing, to what should be a protein. Okay? So when you look at proteins, of course, it's not just the sequence of the protein which is important. This thing, as Purnanand again explained, adopts a three-dimensional shape, which is what actually gives it the function that it can carry it, right? So if you look at these proteins, they come in a wide variety of shapes and sizes. And they are, they play structural roles, they play very critical functional roles. So they are the molecular machines that drive entire life processes forward, right? However, unlike the previous slide in which you saw that there is a code where you can go from the DNA sequence to the protein, a similar code is not there where you can, just look at the polypeptide sequence and say this is the shape that it will adopt. So people thus far have spent a lot of money, time, and effort using experimental techniques such as X-ray crystallography, nuclear magnetic resonance, cryo-electron microscopy, to name a few, to determine the structures of these proteins which play a wide variety of functions. But when you look at the protein molecule itself, it is a linear polymer made up of amino acids. There are 20 amino acids, which again, Purnanand in his master classes already covered. And if you look at the amino acid itself, it has an amine group and it has a carboxyl group. And this part here, which is labeled R or the side chain, there are about 20 of them, which are shown up there. So what happens is that they can covalently link again to form a linear polypeptide. And this is a condensation reaction, which involves the elimination of a water molecule, right? So you have two amino acids, which, condense, and then you form this bond, which is called as the peptide bond. And this is again a partial double bond as. So when these are bonded to one another, these amino acids are called as residues. And the sequence in which you go from the N terminus to the C terminus, so these are different amino acid residues, aspartic acid, asparagine, arginine, cysteine, and so on. And this is called the sequence of the primary structure. So the sequence in which the amino acids are present is the sequence. When you look at the structures themselves of these proteins, it again follows a hierarchical nature. So you have here the primary structure, which is the protein sequence. You have secondary structure, which are alpha helices or beta strands. You have then the packing of these in three-dimensional shape, which is called the tertiary structure. And then further, many such subunits can come together to form the functional unit or the quaternary structure of this protein. in several cases, it could be a monomer, so the tertiary structure is also the quaternary structure. So what is the problem that people are have been trying to address, right? But before I go to the problem, the other thing which is also important is that proteins, again, apart from the polypeptide chain, they can also bind other molecules. They can bind DNA or RNA or lipids or several small molecules and metals, and these can be permanent binding or transient binding. So for example, what you see here is the heme bound to the hemoglobin molecule. One of the biggest insights that has come out of all this genome sequencing and protein sequencing and bioinformatics is that when you look around all the different life forms, these have all emerged due to evolution, right? And they share similarities. So this similarity can be mathematically at least obtained by aligning the sequences of the same protein from different organisms. And why is this important? Because although the sequences may diverge significantly, the shapes of proteins, and this as determined using experimental techniques like crystallography and NMR and cryo-EM, has been shown to remain reasonably similar. And what you also notice is that the conserved residues are the ones which confer both function as well as structural stability of this thing. And again, as Punnanan mentioned, the variations there, you see literally trillions of proteins now from, that's what people have estimated from the various life forms that are there on our planet. And you can start looking at how conserved these residues are. From a computational viewpoint, when you think of a polypeptide, so I just explained to you that you have a linear polypeptide that is a primary structure, which is a sequence of the protein. So if you now think of this, each of them as a ball in space connected by a rigid rod, there are only 3 parameters that are required, essential and sufficient parameters required to describe a polypeptide chain in 3-dimensional space. One is the bond length, that is defined by the distance formula. What is the distance between this point and this point? The other is the bond angle, which is between 3 atoms. So what is the angle that it makes? And the third is the torsion angle. So if you look at this part here, and when we look down this bond BC, what we are looking at is that what is the angle that is subtended when you look down. That is, so if you look at this as BC, and if I look down like this, what is this angle, right? So that angle is called the torsion angle. So if you now look at the folding limitations of protein chains, that is, you have essentially a long linear string which you are trying to rotate at different points and fold it into this is like an origami in some sense, but of proteins. What you see in the upper right corner is a dipeptide system, where when you see those pink arrows, I mean dotted lines, those are clashes. So when you turn, yes. No, see, between every 2 amino acids, there will be 1 peptide bond. Key parameters, right? That torsion angle, bond angle, and right. So in that condition, if I consider like a peptide bond, which is a 5 peptide bond, suppose for instance, then the structure of that peptide bond is possible in a 3-dimensional. No. So we are looking at, okay, so the peptide, you can consider the peptide bond also. In case of the peptide bond, what would happen, because it is a partial double bond, the rotation would be very limited around the peptide bond. So, but other than that, there is no other major problem. So these are the only 3 parameters which are required to define the I mean, I don't know if it is like, if there is a major in the cutoff is decided that at least the protein should have 10 peptide bonds or 20 peptide bonds, then only make a structure. And well, even peptides are can have I think even shorter ones can have structure. So I think, but typically a polypeptide, so the boundary between what is defined as a peptide and what is defined as a protein is fuzzy, but typically very short proteins like zinc finger proteins can be as little as 25, 30 amino acids long. So when you look at this, you see that, when you turn that, you are having clashes, right? So these are essentially, around these bonds, when you are looking at these rotations, you can have the side chains hit into the main chain or into each other and so on. So these are things which you have to avoid. But purely from a main chain, I think Professor G. N. Ramachandran, actually understood this thing in depth, and he actually showed that what you see in the right-hand bottom here is the Ramachandran map, that there is a limited space in which the polypeptide, it's not an infinite space in that sense. It is, I think, but nevertheless a very large space. Like what, I think there are many forms of infinity, right? And this is. So what is the folding problem? Protein folding problem, again, I can save some bit of time here. Because Poonanand has already explained Anne Finson's postulate. So where he looked at the RNA structure, denatured it, and then renatured it, and showed that the structure and the function function is retained, and therefore the structure is and he also talked about what is known as the Levinthal's paradox. That is, if you were to actually look at all possible, sample all of conformation space, it will take you more than the, time that the universe was, which people have predicted the universe has been formed, right? So with these two things in mind, what this implies is that if you are given a protein sequence, all information for folding it into its final three-dimensional shape is already present in the, and this led to a race for trying to think. So this is like trying to find the code of, like the equivalent of the genetic code for protein, folding. So the first model was that of alpha-lactal domain, which was predicted using the X-ray structure of lysozyme. This was as early as 1969. In 1971, actually, people once a handful of structures using X-ray crystallography technique became available. Helen Berman actually started the protein data bank and started urging people from around the world to deposit the coordinates as well as the X-ray data corresponding to these proteins in this database, so that everybody else can openly access and use this. You will find, of course, that this kind of data sets have been immensely useful and are the, backbone, pun intended, to say, of the polypeptide prediction tool AlphaFold. Other tools such as Rosetta, Modeler, and so on, but largely speaking, there are two classes of prediction methods. One is called as the template-based modeling, and the other is called as free modeling. So what is template-based modeling? So you have your protein of interest for which you want to predict the structure. So you search for sequence similarity in the protein data bank and ask, are there any other structures that have already been determined experimentally to which my protein shares a significant sequence similarity? If yes, then I can say that, those regions where it doesn't share similarity, you can, change the amino acids to a thing because you already know how these amino acids look like. But large swaths of structure of the sequence, when they are similar, you can just take that and transplant it. Right? But this is always not the case. There are cases where no template is available. Then you have methods which are called as free modeling methods. And these are based on the physics of the, polypeptide that is based on free energy considerations and everything. The more recently, the methods which have come out, which include AlphaFold, ESM-fold, and are new hybrid methods. And there are also, of course, other hybrid methods like this method Bhagirat, which is from IIT Delhi, from Professor Jairam's group, and many others which coming to the point, AI-based methods have dramatically improved protein 3D structure prediction. And if you look at AlphaFold itself, the first instance of AlphaFold came out in 2018. AlphaFold 2 came in 2020. And AlphaFold 2, when it came out in 2020, was known as, was recognized as the method of the year by Nature methods. And people who have been traditionally determining structures and things, first there was disbelief that something can be so good. And then, they have also started using it to determine or to face some of the structures which they could not otherwise determine from their lab, and started to appreciate the importance of course, it also won the Nobel Prize. But why was the community so keen to latch on to this and say that this is really a path-breaking? This is in large parts to do, of course, one with the protein data bank itself, which has been in, I think, from the 70s, where a lot of structures are now available and people have deposited them. But also due to another experiment called CASP, or the Critical Assessment of Protein Structure Prediction. So one of the, I had into 20, I was part of the evaluation of CASP 5 and in the fold recognition category, and this thing itself has come a long way in terms of what one can do in terms of prediction. So one of the things with AlphaFold is that it has very well established metrics for showing that you have actually achieved what you claim you have achieved, right? And one of those things is called as the Global Distance Test, or the GDT score. So this is a metric where, so what CASP essentially does is that they labs, experimental labs, which are determining structures. John Mould, who is one of the organizers of CASP, tells them, please hold on, don't publish your thing just yet or put it in the PDB. Wait for a couple of months and just give us the sequence, right? And then the sequence is given to the predictors. This is a competition which is held every 2 years. And then they are asked to predict what the shape of the proteins will look like. Apart from that, they also have other things like predicting just the contacts and so on. And why I will come to that part shortly. So the what one could see here is that with AlphaFold 1 and 2, the quality of the GDT prediction, the closer to 100, but closer to ground truth it is. And the minute you go past 90, it means that you are as good, as an experimental structure. About an Armstrong deviation is what you are looking at, right? So how does this program work? So what is the, I think I'll try to keep it simple because this is a diverse audience. But if anybody is interested in discussing this further, I'm available. You can come by. So essentially, AlphaFold works with a very simple concept in mind, that we looked at the multiple sequence alignment of proteins. That is, when you look at proteins from different organisms, you can align them and you can look at which residues are conserved. And the thing is that when something is conserved, it may mean that it is structurally, functionally important. And it may, so if you look at a protein and you see there is a charged residue here and another one of a different charge there, which come close by. If in the multiple sequence alignment, you see that, in this place, there is a hydrophobic residue and the other one there is also a hydrophobic. So there are two key things here, which are modules, one called the Evo former and other called the structure module. I will just discuss that a little more in the coming slide. So what this program does is that when you give a protein sequence, it will run it through a genetic database. So this is your input sequence. It will look for similar sequences from other organisms in a variety of databases, including the PDB. So when it finds a hit in the PDBs, this is called the template. So it makes two different types of files. One is a distance matrix, that is this what is called a sparing here. This is a distance matrix, which is the same as what is being shown here. And the other is the multiple sequence alignment. So from the multiple sequence alignment, it can glean insights as to which residues are conserved and to what extent in the family of this protein. From the distance map is the same sequence on both sides, where you are just making a matrix Ij, and you are asking what is the distance between this amino acid and that amino acid in the sequence. Because the evolutionary information tells you that these things are conserved, you are making a guess that yes, if these are conserved, these are likely to interact. But of course, there may be different things which are conserved. So the question is, you have to try a variety of these. So this goes. So there are two blocks. One is the Evo former block, and the other is the structural module. So this is based on what is called as a foundation model of machine learning called as transformers, which is part of the neural network scheme of things. And the most popular version would be ChatGPT, where it given a token or a question, it looks like you are getting seemingly that you are holding a conversation with the thing. So the same technology, but however it is changed for when your input is a sequence of proteins and your output is the three-dimensional structure of the protein. But what is happening here is that it goes through this. So the cycle of distance map thing. And just to give you an analogy, it is like solving a large jigsaw puzzle. Again, Purnanand showed you, when he showed that, I thought, okay, my job is become easier. No, it is like a large 3D jigsaw puzzle where you are trying to put things together. Maybe you make mistakes, then you remove that part, and then you try to put it back again together. And this is exactly what the machine learning tool is trying to do and determine it can also write out a confidence metric on a per residue level, saying that this is the confidence with which I can predict this. But of what? No, so what you are trying to do is you are only trying to predict the structure of your input sequence. Okay, so one of the data which it takes is the multiple sequence alignment. There are a lot of different things that happen inside the box. For example, if A is interacting with B and A is also interacting with C, then it means that B and C cannot be very far from each other, right? So this is called as a triangle inequality. So these kind of things are going under the hood. But what it is doing is that it is using the information, it's cycling between these two layers. That is of it makes a prediction which may not be 100% correct at the first instance. Then it cycles back, and then it looks at the multiple sequence alignment again. Then it comes back, and I think this block, it goes 48 times. I think it can go more also, but they have shown in their paper that 48 is a good number, where it actually, for a majority of things, it can predict the structure. Then the structure module is a thing where it can, it will predict what they call as a residue gas, or a thing where it just predicts the CNC alpha of the triangle of these things in space. And then it just moves these pieces around to again make the best sense of it. Or to put it in other words, you have the sequence, you think this is how the structure should look like. So the thing is that why it is able to do this, because it has been trained on about 200,000 protein structures which are been deposited in PDB, and you have some idea as to how this, right? And then it will give you two things: one, alignment error, and as well the other is a residue level. So that is AlphaFold 2.
Based on the structure, how the low confidence and high confidence level of the
Okay, so if it is fluctuating too much and is not able to predict it properly, so there are, well, so what it is doing here is that it is over cycles, it is trying to predict. It is seeing which ones are staying relatively invariant, which are moving a lot, and then but there are also other interesting things such as disordered regions, which interestingly AlphaFold people have shown that the regions which correspond to disorder, it actually just goes all over the place. And it seems to be also a side effect is that it also seems to be a good disorder predict. Right.
how can we have the high confidence in the structure that yes, it is the structure, final structure or protein?
Well, it is a prediction software. If there are regions which are poorly predicted, then it means that the program has not been able to predict it. So it is not the equivalent of experimentally determining the structure, right? So it is giving you an answer, but it is also telling you in these parts, I am more confident. In these parts, I am not so confident. Right. Of course, see, the thing, what I explained to you so far was AlphaFold 2. Earlier this year, a new version of it called AlphaFold 3 came in. And this, what AlphaFold 2 could do was only monomeric instances of proteins. That's what it was trained on. It didn't put in ligands. It did not, any interaction, even across proteins or protein DNA. None of these could be done. And these were, there was a lot of harsh criticism that it can't do this or that, because once you do really well, then there is an expectation that you do even better. And so the more recent version, actually, the logic of it is slightly different. It now uses a method called the diffusion model for prediction of the structure. This is AI thing where, let's say you have an image and you add noise to it, and then you are telling the AI that this is a noisy image. Can you get back to the original? So this is something which it uses for training. And so this is available online. To address Purnanand's question, people have actually looked at it. That is it just a pattern recognition software, or is there more to it? That is, can it predict novel folds, brand new folds? And it appears that it can, because gives more emphasis to local rather than thing. And it seems to be, that the AI software seems to have learned from the interactions and it can actually do that. The impact of AlphaFold. So of course, this is from Google's website, so take it with a pinch of salt. So they say up to 1 billion years of research saved and thing. And so the AlphaFold 2 paper itself has been cited about 29,000 odd times, and which means that thing. And our group has also used it quite extensively. A lot of interesting work done with Ramya's group in trying to understand proteins from the gut microbiome, which bind a particular glycan and so on, and several other projects. But since I am mostly run out of time, let me just end with a couple of slides. So one can go you can sequence DNA now, a reasonable cost, and the time for sequencing has reduced significantly over the several decades. When you look at structure solutions, this used to be a thing. Structural genomics and other efforts helped get the lot of structures which are thing. But then AI-based methods which have learned from this can now give you at least a starting point. I'm not saying that these structures are a one-on-one replacement for experimental thing, but you are better off knowing this additional piece of information. So see, in some sense, the paradigm of sequence structure function has changed. When Perut's group determined the hemoglobin structure, they had buckets and buckets of blood. They knew what hemoglobin was doing. They knew the function, right? They were more keen as to how it is doing what it is doing. But molecular biology and other things have changed this thing. So for a long time, we knew the sequence because you could sequence it and there were a lot of proteins, hypothetical proteins and so on in the database. But now we can also have a reasonable idea of how they look. And so, of course, the function is something which, again, probably AI will help bridge. But I was looking at some recent interviews with Demis Hassabis and John Jumper. So what they predict that in the next 10 years, they should be able to model pretty much all protein-protein interactions and things within the cell. So when you have to try to understand life, so what you see here in the left is a list of auto parts. And what AlphaFold can do is to tell you that these parts look like that. Now, how are you going to, what is the blueprint of life? And essentially you made that car. Then how does it go on your road? How does it go when it is in Chandigarh? How does it go when it's in Mohali? How does it go when it's in San Diego, right? And I think this particular AI-based technology has opened up huge, opportunity for researchers all around the world. And so it's definitely, something which is well deserving of the Nobel Prize thing, because it maybe it has not given you the same type of thing as to that the triplet code telling you that this thing, but for a majority of proteins, when you feed it through the thing, although it looks like a black box, it is telling you what the shape looks like, and that is immensely helpful. Thank you again for. Yes, AlphaFold 3 in particular. So after the success of AlphaFold 2, Demis Hassabis, who is also the CEO of DeepMind, opened another company called Isomorphic Labs. So AlphaFold 3 is actually released. It's a joint release of Isomorphic Labs and DeepMind. And their primary idea is to speed up drug discovery because AlphaFold 3 you can, unlike AlphaFold 2 and you can explicitly model ligands and other, multimers, DNA interaction, RNA interaction, post-translational modification. The AlphaFold 3 code itself is now available about 10-15 days ago, they have released it. It is also available as alphafoldserver.com. You can submit about 20 jobs, but the server doesn't allow you to add. So they have a set of ligands only which you can do. But if you have your own ligands, you can give it the smiles thing, or you can define the ligand, and it is possible to model it. You have to, however, sign a agreement with Google when you do that. But yes, it is being used for things. So they also have tie-up with Pfizer and a couple of other companies. So they are claiming that they may be able to bring the next drug into the market in the next 5 to 6 year time, speed it up so drastically. Probably not. The idea there is to to which part of your drug discovery pipeline you are applying this to. So it will be able to go through and look at things. So again, this version this has been released only 10-15 days ago for the public. They have a server which was released in May this year. But I don't think, so what they claim in the AlphaFold 3 paper is that it is a bit more accurate than AlphaFold 2. Although the underlying architecture is slightly different, but yes, it has definitely, opened up a new way to try to understand protein structures, and it has a lot of applications, including in drug discovery. But one has to watch carefully and look at the literature. I can't just say that yes, it will be 100%. Then nothing is 100% accurate all the time.
So I found that some patches are matching. So while already being at living at different temperature and pH condition, so the function will be the same. Or second, can we see in future one option in this AI predictive tool having physiological column saying pH and the temperature kind of things. Like in cell we have chaperones and pH which play a function in proper folding. But in this model, we don't have such kind of parameters. So in future, can we see that at this pH we will give the range and then the model will be this and
Okay, so first thing, AlphaFold is a protein structure predictor, not a function predictor. Second, it is learned from structures which are there in the PDB, and it can do remarkably well, but it can't answer everything. So
37 ਸੈਕਟਰ ਤੋਂ ਆਉਣ ਵਾਸਤੇ
there are
ਬੰਦੇ ਨੂੰ
going to be proteins which
ਰੂਟ ਜਿਹੜਾ ਪੀਜੀਆਈ ਵਾਲਾ
like
ਬੈਟਰ ਹੈ ਜਾਂ
cis
ਕਿ
which can
ਨਵੇਂ ਬੰਦੇ ਨੂੰ ਕਿਵੇਂ ਕਰੀਏ
when they function they
ਵੈਸੇ ਤੇ 37 ਤੋਂ 38
sort of,
38 ਤੋਂ
there is a strand invasion, and so depending on what you are think it is going to give you one view of the structure. It is not going to tell you everything about a protein sequence, structure, and function. But definitely, this is not my work, and this is, but my opinion, very phenomenal work which has changed the entire field.
What a little premature.
No, I think it is.
Another 2 years.
I feel that people always have this thing that experimentalists always have the thing that theory can't think. But I think this is definitely something where, given a handful of structures, if you are able to make a reasonable call as to how other things would look. And again, the problem is that experimentalists, I don't mean all, but generalize, the thing is that you wanted to answer everything that is, possible that protein can do. But here we have to take it for what it is. It is a, tool given a sequence. Most of the times it will give you a reasonably accurate three-dimensional shape. This was not possible before. And as I showed you from the CASP data, the improvement is phenomenal. And this, where the field has been stagnant for many decades now, this thing is a breath of fresh air. And I think it is, in my opinion, definitely worth a Nobel Prize. And I hope these guys, continue to release their code and make it open source as they have done with AlphaFold 2. AlphaFold 3 is slightly less open source. See, the code is out there, but the weights, which is the main thing for the inference part of the thing, is proprietary. But over time, others are likely to reverse engineer this but it has definitely galvanized the field. More people are and it has had impacts in many aspects of bioinformatics. Like improving the quality of annotation, helping find remote homologs, to name a few. But I don't want to take away whatever time is left of Veena. I think director warned me that you may speak longer than Purna. So I think I have done that.
I spoke 27 minutes.
Thank you, sir. Please give him a big round of applause. And I request Professor Purnananda Gupta Sarma to kindly give him a flower pot as a token of gratitude. Thank you, sir. I request Dr. Nishima Wangu to please introduce our next speaker.
So we have now come to the second talk for today, which is by Dr. Beena. Dr. Beena did her PhD from the Molecular Biophysics Unit, IISc Bangalore in 2004. She later joined University of Massachusetts Amherst, US, and as a postdoctoral fellow and worked there till 2011. She joined IMTech as a Ramalinga Swamy Fellow and a senior scientist in 2012. Her present position is that of a principal scientist at IMTech. Her research interest lies in understanding protein sequence folding, structure, function, dynamics, both in vitro and in vivo, using biophysical tools. And today she will be discussing the other half, I should say, the first half of the Nobel Prize in Chemistry 2024. So with these words, I invite Dr. Beena for the talk, please.
I want to thank Nishima for the kind introduction. And at the onset, I also want to thank Mr. Grover and Dr. Khosla for giving me this opportunity. And I echo with what PJ and Krishna said, that this is a topic very close to my heart. So definitely, one of those people who got too excited about the Nobel Prize coming to AlphaFold and the Baker Group. So when Mr. Grover asked me, I was like, yes, we have to do this. We have to tell people, because for a change, when people ask you what you do and you say protein folding, half the people like, so what? So what I'm going to try to do in my talk is, this is a very esoteric field. Like Krishna said, there's a lot of details, even I say, or some of which definitely I don't know about the computational part. It's hard to understand. The point that only a set of people can do that itself is this is difficult. Okay, So since they are students, I want you to tell you what it has allowed us to do. The obvious questions are now over. Should I spend a PhD solving the structure? Probably not, because the structure is already there. Now is the time that we have to ask bigger questions using the information. And one of the questions that was asked, is this 100% rock solid structure solved? That's not true for a crystal structure either. No, this is one of the structures that under a certain condition is a stable structure. So you always have to take that information with a pinch of salt, like any chemical system. Under a certain condition, this is what it looks like. Okay, and then we have to move on from there. Okay?
Slide.
Yeah. So I want to start my talk by introducing you with a brief bio of David Baker. So David Baker started with a BA in biology, studying sociology, and at Harvard. And then he went on to do his PhD with Randy Shepman in UC Berkeley, where he looked at protein secretion pathway, protein experimental. He worked with Dave Agard as a post-structural fellow. And one of the brief points that PJ mentioned that, we think that all proteins fold using a thermodynamic hypothesis, but there is a subset of proteins that also undergo folding using the kinetic partitioning model. Okay? So his work on alpha-lipoic proteins was the first and still remains a classic example of where kinetics plays a role in defining what the final structure would be, which is less stable than what the global minimum structure is. Institute of Protein Design is practically what Baker and his idea and his lab and his group of collaboration is all about. His association has with so many departments immediately tells you that this is a very collaborative work that he has engaged in. The number of papers, patents, companies he has founded, and the people who have worked with him have become independent investigators tells you the breadth of work he has done and also about his personality to some extent. It's impossible to cover all that work. So what I'm going to do is tell you the second half of the problem for which the Nobel Prize was given, how the de novo protein design began, the Baker Lab research itself, and then why do you care? And that example cannot be given better than showing how the Baker Lab, how quickly they worked to come up with molecules that would take care of the COVID infection. Finally, what the challenges for protein designers are, and then some concluding remarks, including what I think as a protein folding experimental person. Okay. A lot of discussion already has happened. DNA to protein. You have about 20 basic amino acids, 2 extra there, but let's disregard that. So different ways of organizing these 20 amino acids gives rise to a unique protein sequence. And for a protein to function, it has to take up a structure or not take up a structure in the cell. Okay? So this sequence of going from an amino acid sequence, going to a 3-dimensional final structure in the forward direction is a protein folding problem. So for a protein folding person, there are 2 things one would want to know. If I have an amino acid sequence, what does the structure look like? The second is the holy grail of protein folding. How does it even get there? Which is not solved yet. Okay? But the first half, like Krishna mentioned, we now have a fairly good way of predicting from the amino acid sequence to what the structure would look like. Look at the numbers. In the open public database, we have about 3,000 million, 3 billion DNA sequences. We have about 200 million protein sequences. The PDB, which houses all the structures that are solved, has only about 200,000 protein structures. That means in the first instance, you would want to know that if I already have certain number of sequences that nature selected, if I can solve the structure, maybe I will get some insight into what they do in the cell. Okay, that's what the first part, protein structure prediction, is all about. It's easy to think about, you go from amino acid sequence to what the structure would be. There's an inverse problem as well, which about 30 years back, at least some group of people started thinking, which is the inverse protein folding. Because the structure prediction was very hard. So let us reverse the problem. Let's say if I have a protein structure, can I predict what sequence will be able to take at that structure? Is there only one sequence? Probably not. It is multiple sequences. If so, then what are those all possible sequences that can take up a particular structure? That's the inverse protein folding problem. Okay. So this also has a challenge. Why? Because like PG mentioned, you take any length of a protein, every position, you have a possibility of having one of the 20 residues. So if you have n number of residues, you would have 20 raised to n sequences. So for 100 residue typical length of a protein, this will amount to 10 to the power 130 sequences. So even, let's say, from AlphaFold, I have all these sequences that I have written down, and I want to predict the structure. This is still an astronomical number of sequences to look for. Okay? But at the same time, nature only has made certain number of proteins that at least we see at present. So all organisms, the proteins that they produce, that number is only about 10 to the power 12. You always have to keep in mind the time of folding, searching, predicting. So all this tells you that if this blue box is a sequence space, then what is already found as native natural protein in systems is marked by these beige color circles. So a few years back, 2018, directed evolution was one of the methods by which people were wanting to improve activities of protein. So directed evolution did not do much. Existing structures, it only made it a little better. So what it immediately tells you is that in this sequence space, there's a large number of sequences that we don't even see. We haven't even explored. We don't even know what they're going to fold into or whether they're going to fold into. This is precisely the inverse protein folding problem that Baker Lab got into. Okay. So how do they proceed with doing this prediction? Two ways. One, you could start with a structure, a known structure, and take the backbone of that structure. Okay. So you're defining the topology of the structure, and then you go on and fit in all the possible combinations of amino acid at every position, and you predict. Okay, you're sampling for every position for the 20 amino acids, and then you look at what sequences are still able to fold into the structure that you have started with. Does it mean that you are always bent on having the structure you started as the only structure? No, you will go a little bit back and forth and make room to adjust and get the final structure. Okay, this was a relatively, according to me, still a difficult but a relatively easy problem. The other one is a much bigger problem. You don't have a sequence and you don't have a structure. So you're looking at this whole blue space and you're wanting to know that from every sequence that actually is possibly existing, are there new structures that will emerge? So the existing number of protein folds that we see in nature, are there more that we don't see? And if so, what are they? So this is an absolute de novo structure prediction with de novo sequences. So as the Nobel Prize also states, these are the structures and sequences that nature has not produced. We have never seen, but the work done by the Baker Group and many others, over a period of time has allowed us to do that. So how does this work? This works because we still follow one principal idea, which started with Anfinson's thermodynamic hypothesis. It says that proteins fold spontaneously. Spontaneity always comes with free energy, and so the final structure you're going to see is a minimum energy structure. So all the calculations that would be done to predict a structure requires an energy calculation. This has always been the problem, because to define a force field that will be able to calculate the total energy of the system, not just the protein, is energy of the system, which has to include the solvent interactions as well. That was the hard part. So with time, and Rosetta was one of the very successful programs that the Baker Lab came up with to predict structures, and it has evolved to a level that it is as good as predicting structure as AlphaFold does, 2 does. Okay, so the energy calculation involves all these 5 parameters primarily, but there are newer models which learns and relearns from the experiment, includes many more terms. Effectively, it's going to look for the LJ potential, that atoms have to be close, but not too close to have sterics. You should have solvation with the solvent system, but you can't put polar residues in the part of a protein which is hydrophobic. Then hydrogen bonding electrostatics, which are involved in giving favorable interactions to fold the protein, and torsional preferences, the torsion angles that both for the backbone and eventually for the side chain as well. So this is the most important part, which actually was responsible for slowing the field, because it's very difficult to get the force field right. There is always an error involved in energy calculation. And therefore, if you think about that, if I make a mutation, if I do something small, will it predict? Probably not, because the error in the energy calculation is not sufficient to do that prediction. Nevertheless, globally, you would be able to get a very good idea about what the structure would look like. Okay? So even though it's an inverse protein folding problem, at the heart of it, you still need to do the prediction. So the Rosetta and other related fields that the Baker Lab worked for many years is still very important, with AI involvement, which they have done after the AF2 code was released, is what led to the success of this method. Yes.
So what about this folding? Like this folding pattern, it can also have a pattern, right? But, so it can be predicted by this way. This folding also.
So a misfolding you have to define. If at this point, using either AF2 or the Rosetta newest models for structure prediction is only telling you I have a sequence, in the end, it's going to look like this. So if, let's say, a misfolding of a protein leads to a structure like amyloid, if that is what you have in mind, then it's not going to predict that. The structure is not going to be predicted. The reason is because, like I said, the whole premise behind this is the lowest minimum energy state. Misfolding always happens because you are accumulating an intermediate which now starts self-associating. Pathway is still not covered. That requires a different kind of an analysis, so it won't do that. So the workflow usually follows that you start with an objective, which would be that the protein that you have in mind, are you starting with the structure? Are you wanting to have a sequence with a certain function to be incorporated? And then you will go back and forth between the structure prediction and the sequence design. Okay. The very important thing that I think distinguishes AlphaFold from the Baker method is there is always a validation involved. That means they always take the final sequence, they express, they would purify the protein, and they will do an assay. That assay would always lead to a structure solving and the function. And then they learn from all, not only what works, but also what fails, and feed that information back to their prediction method. And this goes on till you end up with what works out for whatever your objective is. Okay?
I have a question of this one.
Yes.
So, in all the provisioning that we've done, either starting from first principles or using existing structures as starting points, we've never, in our own experience, have had a success rate of more than about 55%. Baker Lab success rate is nearly 100%. Is that because they only report the successes, or is it because they have such large numbers of people, doing this brute force recycling and reiteration, from result to planning, that they achieve such high success?
No, I think this is what Baker himself always says. The success rate is never 100%. What gets reported is what is successful. I will just point out a paper where they have shown that how much is the success rate. So it is never 100%. One of the papers that got published this month says that it is 99% failed. What you're seeing is only what has worked. But nevertheless, for so many systems, you're seeing a lot of success as well. So I think the problem or the appreciation should be that you're actually looking at sequences that don't exist, and you're still coming up with what it will fold into and incorporate function and what its application is. So, I mean, short answer is that it is never 100%. It is much, much lower than that. They do have a way of making large libraries. So part of, why they still hit upon that, few percent success is definitely because of the scale at which they do the experiment, which we never can do.
Yeah. So we also don't work with totally new sequences.
Yes.
Maybe
yes.
from nature, so maybe it's 60-50%.
Yeah. Yeah. So I think the distinction should be very clear. You know, we're looking at a totally different space here, you know. So the de novo protein design, you know, one of the very famous or successful examples which gave a lot of hope came from the Grado's group when Regan actually managed to fold or successfully create a 4-helix bundle protein. So for students, you know, they start with a 16 residue helical peptide, which could self-associate. Then the problem of connecting the helices, the loops were designed to create a, you know, a 2 helical, helix turn helix, which could assemble still as a 4 helical bundle. And then finally, they were successful in creating this 4 helical bundle. How do you know that it is actually done? So the computationally designed sequence was actually expressed. And you know, one of the methods that is used, because it's helix, of RUV-CD allows you to look at what the structure is, at least the secondary structure is. So you can see that it's clearly a helical bundle. What's also important to note is that when you look at stability measurements, that is, you know, how much of a chemical like a guanidinium you need to unfold the protein, you can see that once you start forming a 4 helical bundle, the protein becomes super stable. The amount of guanidinium which is reported on the x-axis is much higher to unfold the protein. This was one of the first examples which came in 1998. Then the totally, you know, computational method to predict the structure. So that's a 16 residue protein from first principle. You know, a lot of learning about the energy calculations came from Stephen Mayo's group. And here they have actually created a 30 amino acid protein, which forms a complex fold. The helical proteins are usually a little easier to predict, but theirs was the first example where they took this 30 residue protein, which has a motif which is a beta followed by a beta and a helix. And the start point was a known zinc finger protein. And the goal was that you remove the zinc binding function, and then you come up with a sequence that will be able to still take up the same fold. And you can, and this was called as FSD1. The sequence was called as FSD1. I just put the sequence of FSD1 with the starting structure that they started out with. And you can clearly see that there is very less identity between the two sequences. So what they had predicted indeed actually folded into the structure that was that is shown as a computed structure. NMR structure solving of this particular peptide clearly showed in the superposition, which is showed at the end, that the structure exactly overlaid and superposed very well with what was designed as well as with what was started with. The breakthrough, so here you're still working with 30 residue. It's a lot of computational power that you would need. The breakthrough came, this was the first paper from Baker Lab, which was a protein which is 3 times the size, 90 residues. It's called TOP7. So TOP7 was started with a fold that does not exist in nature. And they went again with the inverse protein folding that I described to you earlier. And they were able to actually get the sequence that was predicted to fold into this complex structure of an alpha beta motif, which is shown in green here. And then, they express the protein, they solve the structure, the CD spectrum showed it is well folded, and the crystal structure, which is shown in red, clearly showed that it perfectly superposed even the side chains within 1 angstrom of the prediction that they superposed with what they had predicted. This was the first example which actually gave indication that, it is still a solvable problem. You would have to do something more, but then at least this method exists for large proteins such as at least about 100 residues. The paper that I was telling about, which I think most of the students should definitely go back and read, is a very good combination of what the computational method will do along with the next generation sequence synthesis. You have to create libraries to screen through. This happened because of new technologies which were able to create, synthetic genes, along with the way they designed this method to measure stability. In this whole experiment, see the numbers. They worked with 15,000 de novo designed new sequences. For control, they have taken 1,000 proteins of the same size. 10,000 mutants they made. They actually measured the stability to validate the method. And 30,000 negative controls they took to come up with principles that would allow you to do this kind of a sequence structure prediction. Okay, and the kind of motifs, the topology that they took was very complex. The simplest is the 3 helical peptides, and then going beta structures are very difficult to predict. A combination of different kinds of alpha beta, and these alpha beta structures are very unique. They don't exist. And this they have done iteratively. And in the whole process, from going from one generation to the 4th generation, the success rate has increased tremendously. Even the most difficult protein fold like alpha, beta, beta, alpha, they still could get some sequences that were able to fold to that structure. Okay? So in this experiment, they did not throw away whatever was negative, what did not work. What did not work was used to actually feed the algorithm to figure out what does not work and should be taken care of. So there are a lot of principles that they have discussed about if you were to do protein design. So effectively, and plus I just put the numbers because the paper states that how much money they spent in doing this experiment, okay? Which is also impressive. So finally, they solved some structure, and it was perfect superposition with the what was predicted for these four.
These are estimated numbers, 30,000, 50,000, because
no, this was actual numbers that it worked with.
no, because you see, if you actually make 30,000, you know, $7,000 in reagents is too little. I would imagine that they've created libraries and then done a sampling and then extrapolated to the total number of variants.
No, they had worked with all these sequences, and then in the end they had created, you know, the natural proteins, the mutants have to be made because that's how you're going to validate the method for stability calculation, right? But the actual proteins that were made for these particular sequences is based on the calculation, the effective ones, right? So it still would be. This is only a 40 residue. It's only a 40 residue.
It's a little like the 2018 prize work, right?
For the directed evolution.
Yeah, where part of it was fart display.
Right.
Where you estimate the total number of variants you have by doing a limited sequencing, not actually sequencing the entire library.
My understanding is this is real. Also because this is a new method, which is an oligo-based synthesis of the whole gene, which is very cheap. I think it costs 2 cents per sequence. We also got it done recently with Paul and myself. It's a very cheap method. So I think a lot of success also came because of this new methods of creating library. Okay? So in this also you can see compared to the natural sequences, you had a large number of sequences with the sequence that are unknown are able to take the same structure and the stability is as you are expecting. Okay. These are just an example. I just picked a couple of examples to show you the power of the method. So what is it they did after incorporating AI into their method? So now with de novo protein that you can work with, we will have this space covered for not just the activity which you want to improve on, but you could also incorporate new activity if you know what you want to do. For example, it's one of the examples that is stated many times, plastic degradation or some chemical synthesis. You exactly need to know what is the activity group, which is required to be put in the protein of interest. And you also want some engineered properties like, it should be a controllable system. It should be modular. You should be able to move parts, and domain-like modular system you should be able to create. And whatever is the requirement and the input you give at the end using different methods, you would be able to get the output. So as an end user, where I don't know what goes on in that black box, this method is available. If you go to Rosetta Commons, you know what you want, you can give in the input and you will get an output. Is the output 100% correct? That nobody is claiming. It is just a good start point, and better to have something rather than have nothing in hand. So it works. So that's one of the things I wanted to point out here. And this slide is just to show you that those of us who love looking at structure, this look like magical. Look at the kind of structure. These are all solved structures. Look, these structures don't exist. These are actually designed by first principles. These are design sequences, and the predicted structures are exact, or the observed structures are exactly what the predicted structure is. At the top, you're seeing the topsin 93 residue protein that they started out with, and different kind of geometry-driven structure design At the far bottom end is the mega-Dalton complex, which was able to self-assemble, which was predicted, and by cryo-EM structure, they showed it to be existing exactly as what it was predicted to be. I'll just come back to this structure again. And now, especially after the Nobel Prize, or even before he got the Nobel Prize, the Breakthrough Prize, there was a lot of, interest in this field. So now you'll see a lot of reviews which will talk about various kinds of structures that were predicted by the Baker lab, which is unseen proteins in nature, but also folding into structures that don't exist in nature either. Okay? This is just a list I just showed to give you a reference. So why should we care? Why was this important? When all this was done, maybe there are a lot of people thinking, so what? You have a sequence that folds to a structure. One of the best examples, I think, is during the pandemic, when they were already prepared with this huge icosahedral nanoprotein particle, and they already knew how to predict the structure. We all went through it, no need to tell how difficult it was. The numbers dropped. These numbers of infection and death dropped because the vaccines came into picture, the antiviral molecules came into picture. So for the reasons to look at what component of the viral structure is important, the schematic show you that, about November, December of 2019, when coronavirus was first detected, the genome sequence came within a couple of months. By the time the genome sequence came, Baker's group was also one of the groups that first quickly predicted what the spike protein would look like. Spike is a primary glycoprotein that is covering the envelope of, or the outer surface of the virus, and it makes the interaction with the receptor, and the primary receptor in humans is the ACE2 receptor. Okay? And from this analysis, it was clear that the receptor binding domain, a small, about 200 residue domain, was responsible for making the interactions with ACE2. Because you knew this interaction and the significance of this interaction to cause infection, it was logical that you have to break this interaction if you have to stop the infection. The other way of seeing is that you should have a vaccine candidate which recognizes whatever is on the outer surface, and probably because spike is covering all the overall surface, you should design some vaccine candidate which is some part of the spike itself. So the subunit vaccine, as well as molecules which will break the spike ACE2 interaction, but designed just because the structure was available before it was actually solved by cryo-EM, by the model structure. So within 2 months, they already had the structure of the trimeric spike. They knew the RBD was the responsible domain of the protein to make the interaction. Subunit vaccine, one could even start with RBD alone, which is what is done. One of the groups in India, Ragan's group, also came up with RBD as a molecule for subunit vaccine. But the Baker group did differently, is that they thought that let's decorate or increase an equivalent of avidity by putting more number of molecules of RBD, and they decorated the surface of this nanoparticle, the icosahedral particle, which is actually primarily the work done by Neil King's lab, and they created this nanoparticle, protein nanoparticle, which carries 60 copies of RBD, and then there's a lot of paper talking about how successful this is in comparison to a single RBD being used for generating the immune response. And this was one of the first nanoparticle-based against COVID vaccine, which was approved. And eventually, because of the way the infection proceeded, like all vaccines, it faded out. But nevertheless, what I want to say is within 1 year, it had gone through all the steps of defining the right candidate and working out the right molecule and getting a vaccine which was already approved and in the market. I think this is very...
but it needs to be also said that coronavirus has been known for decades, and various versions of the spike protein have also been known.
But they're very different. They don't work the same.
Yeah. So there are variations, but a certain amount of homology-based modeling must have also played a role here, not de novo modeling. Because all these spike proteins from different coronaviruses have
probably right.
high homology.
No, but let's say if I take SARS-CoV-1 versus SARS-CoV-2, whatever raises immune response and the antibodies that are generated against CoV-1 will not recognize CoV-2.
That will always be the case because of the microenvironments at the antigenic site at the epitope being different.
True.
I'm saying for modeling the backbone trajectory, some
Possible. No, true. But my thought is that, we also had it. Did we do it? I think some part of that realization has to be there that, maybe it looks trivial, but it still happens. Those who did it and went forward is, I think that thought is always there, right? The other thing is here, they have decorated the nanoparticle with it, which made it so much better than even the antibody, the regular whole protein, spike protein mediated response as well. Okay.
Just as an older person and also nominally as guest of honor, it's my responsibility to make it a little more conservative when the hype is
the one. Oh, no, I'm okay with it. See, I will quickly go through the time, but they also came up with the antiviral strategy, wherein they actually looked at small molecules that are, derived from the ACE2 receptor, and they were able to, all this work is within 2 to 3 months of the time that the infection was detected, and they already had the models, and these molecules are binding the RBD, the ACE2 derived mini proteins are binding to ACE2 with picomolar affinities. So they solve the structure. The pipeline, once it is set, then you have the molecules. But what they have done next, because the variants were coming up, they took these molecules and they created these, fused them and created the dimer and trimer kind of molecules. And this was the other antiviral nasal spray. I couldn't find what happened to it eventually, but then this was also successful, at least in the animal models. Okay. So this was just an example to tell you that it was important to be ready with the technology when it is needed. And that was only because, you could do all this kind of protein engineering and protein design work. Okay. So in 2019, before COVID hit, Baker gave a TED talk, and then he defined what they are going to do. And there were like 5 challenges that they had set forward with: forming protein nanoparticles, new therapeutics, that delivery, vehicles, smart therapeutics, and next generation materials. And 5 years down, you can go back and check, and practically in every area they have come up with a molecule. Okay, so they have molecules which are for targeting specific cancer, molecules that will take up small molecules like antibiotics and deliver to the site that you want. You have molecules that are like, even building in chlorophylls into a different framework. Fluorescent molecules, which are beta viral proteins, which are very hard to predict with different sizes. So that will allow you to do DNA sequencing, protein sequencing. These are all design molecules. The advantage here is that all these design molecules have very high thermostability, which is a big plus for applications that I just described. Okay. Does it mean that everything is done? No, absolutely not. So there are challenges which are still existing. Like, PG has pointed out, is the success rate 100? No, it is not. So these are systems that we will need to build it. So for example, predicting binders, catalysis still is a very big challenge because catalysis requires very small changes. And if you are a couple of angstrom off, it fails. It will not work. I can't have a protease with a couple of angstrom. It's never going to work, right? Conformational changes, dynamics which are intrinsic to proteins to work, those have to be incorporated. Very complex reactions, building motors like a flagella, these are systems that are very difficult to make in an artificial system. And then most importantly, the success rate itself. You're going to have a lot of failures, molecules that don't work, right? So you have to learn from them and then go back to the model, and then probably it will evolve to be better. Okay? This is my conclusion slide. So, the sequence space, structure space, and the function space in natural protein, because of evolution, the sequence space is definitely fixed because I have a DNA sequence, I know how many protein sequences. Structure space, because we don't see all kinds of structures existing, new folds exist. Function, it's a requirement based. Today, a cell has a particular requirement, it's going to evolve a protein that will do that function. So why don't we see everything that theory predicts? So is it that nature has seen it and realized it doesn't work? So now, those are ancestral, so we don't see it. Or, it has never been required for nature to produce those proteins. So what this de novo protein design allows us to do is to be able to capture that sequence and structure space. You also need to remember that when you think about natural evolution, it's pretty well established now that this protein sequences are mostly evolved to perform function first before stability. Thermodynamic stability always comes after the function. So probably those functions were never needed. Why this is important is because problems of, let's say plastic degradation, billions of years that probably never required before plastic was involved, came up, right? So now these methods will allow us to actually take care of our problems in a much faster way compared to waiting for evolution to come up with a solution, right? So initially we were always, this is all a process of evolution. So you go from natural evolution to directed evolution, which happens on a lab bench. Now, because of this, tremendous leap in how the in silico protein structure prediction has happened, you can go back and forth. So scaffolding is one of the places wherein, you already take what is required, you put it in, and then I think what Krishna briefly mentioned, wherein, you are working in a sequence space, and then you do this noising and denoising, which is a very cool technique, and you're coming with totally new structures. I think that's very exciting. So large number of methods have allowed us to come up with these de novo protein sequences. What we learn about de novo protein sequences is that they're very similar to natural sequences. Therefore, but the stability almost always is more than what you observe for the same fold in a natural system. They can form novel, nanostructures. They can also perform a lot of functions. Right? Catalysis still is a challenge, but nevertheless, we are getting there, because even in evolution, you start with a poor performer, you evolve and get better. So there is still hope that if I get a poor plastic degrading enzyme, I can just make it better by using multiple methods. Okay.
In the mid-1990s, experimentally, someone tried to make 50, 60 amino acid long constructs combinatorially.
Yeah.
So every level of condensation of an amino acid derivative, they would use all 20.
Okay.
So they built up, I think, a few 100 million or something, different sequences, again, from extrapolation, so we limited sampling. And they found that less than 1% of those were folded. So that means that in that vast sequence space, most sequences don't actually have a three-dimensional unique structure.
Possible, which is what, but at least, knowing what is not feasible is also important.
But when you ask that interesting question, has nature sampled all? Right on the right top, the previous slide. Only in a steady state universe, would it be possible? Certainly not. Certainly not in this 14 billion year old Big Bang.
Probably, yeah. So I think all this became possible because of open science. I think that to me is the most important thing. This field has evolved because of open science. PDB, like Krishna said, 1970s. I think it was, these methods work because all the structures are deposited, and we have learned from them. And people who have worked in the field has also contributed as openly, and both AlphaFold and Rosetta were always open, And the other thing important, I think, from this particular Nobel Prize is, how interdisciplinary it is, and how integrated the work had to come together to give rise to such a big breakthrough, at least according to me. I think this is very important. And for students, I know when myself have looked at it, this is a very cool, game. It's a folding game, which you can actually look at. You will learn a little bit more. It gives you scores. It is quite folded. And if you're interested in the field, you are excited about the field, you can look at, a lot of information shared by the Baker Lab in a community which they call as Rosetta Commons. So there are, they have training programs. They also have, a lot of educational videos where you can learn. They also share all the codes. So those who are interested in that aspect, they can also look at it. So I think I'm going to end here. Thank you for your attention. So this really led to a lot of useful information or useful stuff for the society. So Baker is also one of the 100, most influential times people in the health category, and definitely the Nobel Prize probably seals it. Says that this is a very important contribution.
Thank you.
Thank you, ma'am, for that informative lecture. And I would like to request Professor Arun K. Grover to kindly present a flower to Dr. Beena as a token of gratitude. Thank you, ma'am. Thank you, sir. With that, I would like to request Professor K.K. Basil from Punjab University to kindly present the vote of thanks on behalf of NASI.
Thank you very much. I would just like to make a mention that this is one of the most interesting talk which I have ever heard. And so I take my duty. Good evening, ladies and gentlemen. I take this distinct opportunity in expressing my heartfelt thanks to highly accomplished and decorated distinguished speakers. I have mentioned the name as professor, but I don't think they will mind because I'm from the university. So they are Dr. Sri Krishna Subramaniam and Dr. Beena Krishnan for their remarkable presentation. Before I came, I was given an understanding that there will be only 2 speakers. But I'm very happy that there are not only 2 speakers, there are wonderfully 3 speakers. And one of the iconic speakers. I'm sure that you all will agree with me that both Dr. Sri Krishnan Subramaniam and Dr. Beena Krishnan expressed in the most simplified way the work carried out by this year's Nobel Prize. The relevance of the insights shared by the learned speakers is crucial for motivating researchers, and I'm sure that the presentation by the speakers has reignited innovative ideas in the mind of our researchers present here today. The most rewarding component of this fantastic talk was the emerging question-answer session, with curious queries from audience that raised very many doubts and suspicions. I think there are still very, very doubts, but I should make a mention. I may add that both the speakers have left no stone unturned in crisply demonstrating through their visionary talk. Please join me in passing a hearty vote of thanks with big applause for these speakers. Special thanks goes to the director, IMTech, Dr. Sanjeev Khosla, Professor Arun Kumar Grover, ex-Vice Chancellor, Punjab University, and their colleagues for playing the instrumental role in inviting and coordinating with today's speakers. Together with continued support, encouragement, and providing the necessary facilities from IMTech. We deeply appreciate their collective commitments and sparing their valuable time in spite of their hectic schedule. I'm thankful to today's guest of honor, Professor Poonanand Gupta Sharma from IISER Mohali. I was given an understanding that he is not the speaker, but he was the most iconic speaker. Thank you. We are especially thankful to Professor K.R. Dharamveer, General Secretary, SPSTI, and the Society for the Promotion of Science and Technology in India for support of these lectures conducted in association with CSIR IMTech, Chandigarh chapter of NASI, INSA, INIAS, QUIC, Chandigarh PI Rahi, Amity University, Mohali. I'm thankful to Dr. Nishima Mangu, who happens to be my student also, so for conducting the stage. A heartfelt thank to Rajiv Kumar, Mahipal Sharma from SPSTI, and Ms. Nandita Sharma from IMTech and their dedicated team who worked diligently behind the scene to ensure the overall success. On behalf of SPSTI, CSIR IMTech, Chandigarh, NASI, and I'm indebted to Professor
speakers, I should say, not professors now, the speakers for being with us this evening with their scholarly, highly enlightening and mesmerizing talk. Thank you so much.
Thank you, sir. That's all for today's program. I request you all to join us for a cup of tea outside the hall. Thank you.
Very nice.